for it.
Okay, there we are.
We starting from the beginning?
Yeah, that would help, wouldn't it? Hi to everyone joining us. We're just waiting for some more people before we get going. Yeah, we're just going to give people a few more minutes to join before we start. Sorry, I just wanted to reiterate for people joining us that we're just waiting for a few more to join before we begin. Okay, I think we might start there. So welcome everybody. Thank you for signing up to attend. And welcome to this presentation on a strategy for managing contamination risk in segregated hospitals. Our presenters today are Tautvidas and Rick. Just waiting for that to move on. So Tautvidas is our head of research and development, and he's an expert in harnessing decontamination technology to eliminate viruses, bacteria, and other contaminants from hospitals, public spaces, and workplaces. He's led research and development into numerous pioneering solutions for virus outbreaks, including isolation pods, automated hydrogen peroxide vapor, and ultraviolet C systems, as well as decontamination wipes. He also serves as a committee member for the British Standards Institute, and he's had his work published in several research papers. Rick Fentiman is our managing director here at Inivos, and he's been at the helm since 2007. His role has given him intimate access to knowledge and evidence supplied by his skilled team, as well as the wider scientific community. So he has extensive knowledge on infection prevention and control, and he uses his management skills to get the very best out of his team and to provide tangible decontamination solutions. And it's to Rick that I'm going to hand over now to start things off.
Thanks, Freddie, and thanks everybody for joining, and it's a great pleasure to talk on this subject. We're obviously in unprecedented times, and it's the subject of segregated hospitals, I suppose, has been a base principle of infection prevention and control for many years, although it's in a much larger and more sophisticated scale now. And I suppose it's today, the purpose of today's presentation is really to talk through some of the experiences we're seeing and being involved in on the ground floor, along with many hospitals, to create safe places to care. Inivos as a business is a specialist focused on the development of technologies and services to reduce the risk of patients acquiring infection in hospitals, I should say. So our focus is very much around the acute healthcare sector and the acquisition of infections in those spaces. Our aim is to enable hospitals to deliver better care faster. So I'm going to next slide, Freddie. So we believe that any infection prevention intervention there should be a structured approach to it, and we use this understand, design, deliver methodology. It's split into 3 segments, as you can see. Understand is very much understanding what is the need we're trying to fix. Design is designing it based on best practice, designing it based around evidence, peer-reviewed evidence and studies. And then delivery is looking at an execution model that actually delivers the net result that we set out to achieve, and monitoring it and measuring those using empirical data. And I think using this methodology really helps us to make sure that when we set out to achieve something, we know whether we have or not. We see too many times healthcare providers implementing infection prevention control strategies or interventions of any description without a clear plan of the end the end from the beginning. And it ends up being a bit of either a vanity project or a something hard to substantiate as its real value. So I would recommend to any healthcare provider and anybody that's working in infection control to really have set out, whenever you're bringing in any intervention, to have a really clear understanding of what you're trying to achieve in the first place. So just can I have the next slide, Freddie? So I think that kind of leads us on to the next point about vectors of transmission, which Tautis will be able to talk us through.
Yeah, so thanks, Rick. And just following on our approach, and saying that before we dive into the areas where the risk of infection needs to be managed within the segregated hospitals, it's first important to understand how infections are acquired. We often refer to these common vectors of transmissions, and these include surfaces, water, air, and people. And if patients came to the hospital and had acquired an infection during their stay, it's likely that they have acquired it from one of those vectors. So we know that microorganisms can survive on surfaces, and so equipment in the hospital built environments, walls, floors, ceilings, etc., pose a threat in the transmission. In water, the transmissions can occur through naturally occurring contamination from incoming water source, as well as through the spread of water waste outlets. Air can transmit infection through air handling systems, or droplets can be spread from one source to another. And infection can also be transmitted from person to person. Next slide, please, Freddie. Within the context of the COVID-19, we see that the surfaces pose a threat, as the virus is able to survive on different kind of surfaces for varying lengths of time. And there's a number of studies that suggest how long the SARS-CoV-2 virus can survive on the surface. And although some information is conflicting, it's fair to say that they all agree that the virus is able to survive on surfaces for a long period of time. So air too is a vector of transmission, particularly spreading the droplets when performing aerosol generating procedures. And so are people, through poor hand hygiene practices or coughing and sneezing. And all of us are, of course, are aware of this, given the social distancing measures that are put in place, globally. And so far, water doesn't seem to, provide a reservoir for COVID-19 to exist, so it's not a vector of transmission. So with that in mind, and making sure that we do understand how infections are acquired, I'd like to hand it back to Rick to talk about the COVID curve. Next slide, please.
Thanks, Freddie, and thanks, Tautis. That's helpful, and I think it's always worth breaking down a complex problem of infection prevention and control, and what more complex than COVID-19, into some sort of key areas to focus on. I think we've seen hospitals do this very effectively in the last few weeks and months. Just looking at the overall kind of epidemiology curve of this outbreak, I think what we're all seeing in the public data is that numbers seem to be flattening. There's no sign of significant decline yet, and it's very hard to predict with any level of confidence how this outbreak will play out. I think one thing that we're seeing, and the politicians are constantly reminding us, is that the data is inherently unreliable, and we're all measuring things slightly differently. And I think we're going to be studying this data really for generations to come, whilst we figure out what we can learn from this data and what we can learn from this whole outbreak. I think in a way it's a certain level of privilege we have in being alive in these days and being involved in healthcare in these days. I think we're in unprecedented times where we'll learn lessons we've never learned before, and probably hope never to learn again. So very interesting days, and we should, whilst it's important we keep safe, there's so many opportunities for learning at this time too. I think the real questions for me are, what does the pan healthcare connectivity, how does that fit with this whole infection outbreak? What impact is, or what level of contamination, cross-infection is happening in the community? What type of cross-infection, cross-contamination is happening in the community? And what activities are driving that? And then how does that play out in the acute healthcare sector? And I think we're starting to see hospitals talking about hospital onset COVID, which is really interesting, because up till now it's been very much about the community playing the biggest part in that community acquisition being the biggest part of that. But now we're seeing hospital acquired COVID start to be a very interesting discussion point. And we're also seeing a lot of discussion around what winter 2020 will look like. And I know we work with hospitals in Australia and New Zealand, and they're not seeing any reoccurrence in any significant numbers, but they stand poised, ready for that in case that happens. And that's not to say that we won't see any reoccurrence in the UK. And I think we're seeing the way that the government is standing back from the whole Nightingale facilities and putting them on hold, so to speak, as additional capacity in case we should need it, is testament to really the fact that we don't really know how winter 2020 will play out. I think the interesting thing too, right at the moment, we're seeing hospitals, whilst there's been this massive focus on COVID-19, and we have seen in the last few days and weeks, hospitals start to see issues with Clostridium difficile and CPE and some of those other more common hospital-acquired infections start to rear their head again. So it's, I think it's really important that we don't lose sight on our core infection prevention and control strategies whilst we're in the face of this current pandemic. Next slide, please, Freddie. Somehow we need to get better at the IT thing, I think somehow. I'm sure we're all learning more about IT. So the subject of segregation in hospitals is a very interesting one, and it's a basic principle of good infection prevention and control. Obviously, segregation of our infected patients and our non-infected patients should run through any infection prevention and control strategy, but we're seeing this in a massive scale in the current crisis that we're faced with. I think we see in the UK, it's very interesting, we're seeing a variation of a variety of segregation models. We've seen hospitals who have multiple sites within their single trust or within their health board start to talk about segregating between sites. We're having COVID sites and non-COVID sites. We're having hot sites and cold sites, red sites and green sites. I'm sure there'll be some sort of consensus on this at some point in the future. And then we're seeing hospitals who don't have the luxury of having multiple sites trying to create segregation within a single site. And that's really challenging because there's so much of our facility is shared and so much of our resources is shared. So that's a really interesting and challenging need within our current healthcare facilities. I think we're seeing a massive increase in routine testing, and I think there's a massive debate about routine testing of patients as well as healthcare workers, asymptomatic particularly, and the needs for that and sort of what drives that. And then the challenges, the logistical challenges of routine testing, particularly for elective surgery patients. So when we've got elective surgery starting to come back online, what's the process for screening of those patients? How will we manage the logistics of their of their swab to get back to the hospital to be tested and the results to be managed, et cetera, et cetera. So I think there's a massive discussion there, and there's lots of work and lots of opportunities for innovation, I hasten to add. And we should be mindful of that. There's massive opportunities in all this adversity. There's innovation too. So we should reach out for those points. Next slide, please, Freddie.
Yep.
I'm sure it'll come. There we are. Just continuing that point. Segregation in hospitals, we're seeing the management and detection at triage being a big discussion. So how do hospitals manage and detect COVID-19 at triage phase in a hospital, and then changes in the way care is delivered, the care delivery methods to maintain social distancing within our healthcare facilities. So social distancing inside the hospital felt like a virtual impossibility, and I think it's been really challenging for a lot of hospitals, but we have seen hospitals reducing bed capacity to increase their likelihood or the possibility of managing social distancing in the healthcare facility. We've seen a massive increase in the use of IT, which in itself, although is excellent, and we're seeing advancements in IT that we've been maybe pushing for the last 10 years for happen in the last 10 weeks. At the same time, it brings a whole new infection control management challenge around how do we manage the potential contamination of those devices, and what's the process there. I think we've seen the restriction of visitors to a level that we've never expected. Whoever of us would have forecasted that a few years ago, or even a few months ago. But wow, wouldn't infection control in a hospital be a different thing, a different animal to manage if we didn't have so many visitors? And I think that's going to be really interesting to see how this plays out in the next few weeks and months around the restriction of visiting and what impact that might have on our infection prevention and control strategies. Next slide, please, Freddie. So I believe in any intervention of any description, especially when it comes to automated decontamination, which is what our particular focus is. We need both proactive and reactive interventions. Now, proactive interventions are interventions we take ahead of a confirmed infection case or cross-infection. So this might be, for example, where we go in and do and perform some sort of decontamination or cleaning activity in a space without really knowing that there's any infection risk there, but we do it because we perceive there may be a risk in the future. And then reactive interventions are when we have a known confirmed infection risk, and we have a known, so discharge of an infected patient is a known infection risk, and I think that's where we would turn to reactive interventions. Now, this doesn't make proactive or reactive interventions right or wrong. It just means that you need to have both proactive and reactive interventions to make it an effective system inside any hospital. And we see hospitals who focus very heavily on the proactive approach, but nevertheless, you can't avoid the fact that if you've got an infected patient in a side room, you need to perform some reactive activities in order to make sure that space is safe for the next patient. So I re-emphasize the point, it's not about proactive and reactive interventions being right or wrong. It's more about making sure that we're using the right approach in a balanced way, depending on the need we have in the hospital at the time. Next slide, please, Freddie. So here's some of the facilities we've seen in the last few weeks and months, and some of the challenges we've seen that hospitals are faced with around environmental contamination particularly. So we've seen testing facilities in varying formats. The pods we saw originally in the early days and weeks of the COVID outbreak, and we saw a lot of hospitals performing proactive decontamination in those areas using hydrogen peroxide vapor and also UVC light. And we saw because there's an unknown level of risk, and I think that's a really important thing to understand here, that when we've got asymptomatic patients going into these environments, there's an unknown level of risk. It's very easy to manage symptomatic patients because they are now a known symptomatic. There's much more clarity around the level of risk that they're giving us. But when we've got asymptomatic patients, it's a different piece to manage. I think the testing facilities obviously have moved on to the drive-through methods, and we're starting to, it'll be interesting to see how, as we come out of the lockdown, as we come out of this COVID-19 curve in the UK especially, and in each of the countries that are on this call today, how we start to reuse the pods again. I think there will be a role for them. I think there will be a place for them, and the investment the hospitals put into those in the early part of this year, they'll start to see it play out again. So I think we shouldn't ignore the fact they have a purpose, and I think we shouldn't ignore the fact they present a risk. As soon as we've got patients going through them and using them in confirmed infections or unconfirmed infections, that presents a risk, and we must make sure that those environments are regularly decontaminated to reduce the risk of that transmitting to another patient. Next slide, please, Freddie. This is snail's pace IT.
Yeah, a bit of a delay, I think. There we go.
Aha, good. So admission areas, we've seen a really big focus on admission areas, both both emergency, and then in the last few days, people talking about the elective admission areas and how we build the patient journey, so to speak, of our patients coming into hospital, if they're coming in for elective surgery, how will that happen? Patients coming in for emergency, for some emergency reason, how will they be managed, and what's the care pathway for them? And then last but definitely not least, our COVID patients, our confirmed or query COVID patients, symptomatic COVID patients, what will be the patient care pathway for those patients? Typically, admission areas of any description tend to be short stay, high throughput areas, i.e. the patients are not in those areas for long, and there tends to be as fast as they've been moved on to their new ward, or to a ward, or into the healthcare facility, the hospital, there's another patient hard on their heels, ready to take their space. And I think we've seen very interesting data for Clostridium difficile, where the role of these emergencies, particularly the emergency areas of our hospital, playing a significant role in the cross-infection or cross-contamination of C. diff in patients. And we've seen some very interesting studies, a very interesting study that was published by Mike Weinbrun. I'm sure we'll be able to share it after this. We also did another project at Bucks Health in the UK, and that was very interesting to look at a proactive decontamination program in their ED facilities, made a massive impact on their C. diff. And we coined the phrase the dirty front doormat, and I think that really describes it, that if our EDs, if our emergency departments are contaminated, if there's a high level of bioburden in those environments, it will transition into the hospital, and we will see it transfer into the hospital. And we spend a lot of time in our hospitals focusing on infection, cross-infection and infection outbreaks on the wards. And I don't believe we spend enough time looking at the route of transmission back down the patient pathway, patient care pathway, back down to their admission route and the front doors of coming into the hospital. So I think that's something I would strongly recommend in these periods of unprecedented lack of activity in some of our hospitals. We've seen massive drop of activity. I'd use this opportunity to get into those hard to access areas and perform deep cleaning and decontamination in a good old fashioned way, to take advantage of this reduction in capacity that we're seeing right at the moment. And we've seen hospitals do that very effectively, and I'm really looking forward to analysing the data going forward, because I really believe it will make a difference to not only COVID, but also the many other hospital-acquired infections that we're very focused on: C. diff, MRSA, CPE, etc. Interestingly, the product you can see at the top of the page here is a fixed UV light decontamination system. It's being launched to market in the next month or so. The idea is that this will be something that we can fix above in a single isolation room, or in a cubicle area, particularly of an accident emergency room, or a treatment room, or any of those spaces. And we can rapidly decontaminate that space post-discharge of that patient. So we're thinking this will typically be an 8 to 15 minute cycle time. So a massive change in the whole dynamics of being able to create safe spaces in our emergency departments and accident emergency areas. So anyone that's interested in that concept is feel free to reach out, and we can talk you through in a bit more detail how that would work. But very similar to the Ultra V as a mobile decontamination system, this is a fixed device that will fix into a facility. We're seeing a lot of interest from the dentistry sector, because dentistry is obviously faced with some massive challenges around aerosol producing or aerosol generating procedures, AGPs, and we've developed this technology particularly with those areas in mind. So any interest, I'm happy to discuss that outside of this presentation. Can I have the next slide, please, Freddie? So single rooms, we've seen single rooms used for both isolating COVID patients and also protecting non-COVID patients. I think it's been it just really depends on each hospital and how they've been managing their COVID crisis. And I think whatever you've been using your side rooms for, I think it's really important that we decontaminate these post-discharge of infected patients. So hospitals, we've seen using it not only is to make sure that there's for a confirmed infectious case in a side room, when that patient moves or goes home or is discharged, whatever happens, we've seen hospitals very effectively taking that opportunity to decontaminate that space to make sure it's safe for the next patient to arrive. We've also seen where you've got immunocompromised patients who have been placed in that space, for the duration of their stay to protect them from any other infectious risk, that we're going in proactively decontaminating that space post their discharge to make it safe for the next patient. So I think it's not only about cleaning up after the last patient, I think it's also about protecting and protecting the next patient that comes in. And I think it's a really important kind of shift in mindset we need to think about. And typically hydrogen peroxide vapor is the methodology that people use in this application, just because of its level of efficacy and how much more efficacious it typically is over against UVC light. However, where you've got high throughput and a particular pressure on time, UVC is an excellent tool for the job. Can I have the next slide, please? Ready? Excellent. So dedicated cohort areas, I think every hospital has been using these for the duration of the COVID crisis, and we've seen hospitals who, during their whole infection, throughout the whole COVID crisis, we've seen them segregate either by whole cohort wards and then whole cohort bays, and I think we've seen whole wings of hospitals being used as cohort areas inside hospitals. I think there's going to be it's going to be a very interesting curve over the next few weeks and months whilst we come out of this current COVID crisis, whilst we see hospitals revert from maybe a positive cohort COVID area back to its normal usage, and then we're seeing hospitals who are either downscaling or upscaling their COVID capacity by creating bigger or smaller COVID areas, and I think cohort areas. And I think there's a massive requirement for using a decontamination technology that makes sure that the risk of transmission of infection between those transitions is minimised. I think we've seen hospitals in Australia, particularly where they've been using outlying capacity in hotels and leisure facilities, where they've been using that as COVID capacity. We've seen a lot of decontamination activity happening in those facilities where we've gone in and done decontamination of the whole hotel block. We've seen hospitals in the UK who are looking at downsizing their COVID capacity, and they're wanting to revert back to its original use. We've had a hospital in the north of the UK who took over their cystic fibrosis unit as their COVID isolation unit. Excellent facility, excellent unit, except for they when came to transition back to a COVID unit, they go back to the cystic fibrosis unit, I should say, away from COVID-19. They had to go through a whole process of making sure that environment was safe to readmit their patients back into. So I think it's very well worth thinking through what risks we might be exposing our normal patient base to, as we come out of this whole COVID crisis. And it's something that we've seen hospitals very opportunistically, and I think very kind of, I think it's a brilliant opportunity to get into there and conduct good old-fashioned manual deep cleaning, facilities maintenance, all those minor tasks that we've been wanting to do in our facilities. We haven't had the capacity to do them. Now is a brilliant opportunity, and we've seen some excellent examples. I was thinking particularly of the hospitals in Wales we've been working with. They've done some excellent work down there, and I really commend them. Happy to put you in touch with them if you're interested. After this call, they've done some excellent work in using this opportunistically to their strength. Can I have the next slide, please, Freddie? So I think the shared patient areas we've seen, which are interesting facilities, particularly because they are exactly what we describe them as. They're a shared patient area where multiple patients are using them for the same activity, particularly shared patient bathrooms, X-ray. We've seen the gymnasiums, treatment rooms, etc. And I think there's a real risk that not only for COVID-19, we should just emphasize that this is not just about a COVID-19 strategy here. This is about making sure that our shared patient areas and equipment do not present an enhanced level of risk to our patients. So we've seen gymnasiums, particularly physiotherapy gyms, where patients going in there back-to-back patient lists, and really challenging for physiotherapists who are busy enough as it is to conduct proper cleaning and decontamination between patients. So we've seen hospitals not only doing proactive decontamination, getting in there on an interventional basis, i.e. once a week, once a month, once a quarter, whatever it is, to decontaminate those spaces to reduce the risk of those transferring any risk of that contamination across to the next patient group. but also seeing hospitals who are saying, okay, if we have to take infected patients to these facilities, we have a method of effectively decontaminating it once those patients have been in there. I think UVC light is a really useful technology for that. It's rapid, it's fast, it's residue free, so there's no residue at the end of the cycle. So it's a very useful technology for shared patient spaces, particularly those that are under pressure and have got back-to-back usage all the time. I think we're going to see an increased use of the fixed UVC light system, and it's something that we're very excited about to see how that plays out from here. I think I'm handing over to you now, Tatis.
Yeah, thanks, Rick. If we can have the next slide ready. So I suppose what I'd like to touch upon as well is that an often overlooked area that we should consider, or we should consider in our strategy for managing the risk of infection, is our emergency vehicles. And then with the emergency vehicles both arriving and departing to the hospital, there's an unknown level of risk. As you know, the vehicles may have visited an area of higher risk. We need to consider several touch points with spaces outside of the controlled clinical zones, such as patient homes. There's also a risk of being close to the contact with the public with their infections and with the coronavirus potentially being, you know, at a known level. And an ambulance crew are equally at risk of potential carriers of pathogens. So although it can be difficult for staff to follow robust infection prevention and control practices, there's definitely a proactive approach, and there's, you know, we can practically decontaminate, absolutely, you know, it's possible to practically decontaminate those emergency vehicles. And you should treat those spaces the same way as you treat any other space in your infection prevention control strategy. So we've just recently had this opportunity to work with DSTL, and that was in response to the open call for a rapid ambulance decontamination method. And we took our UVC system there and just literally tried to work out, you know, what was exactly the, you know, the problem. And basically they were faced with the fact that once the ambulance would, you know, would carry the COVID positive patient, they would have to go back to their cleaning station to try and do the, you know, the clean to make sure that, you know, the ambulance is safe for the next patient. And that was taking a lot of time. So not only that we thought that perhaps we, you know, we could obviously use our existing product to decontaminate the space, but we thought that, well, we were thinking previously that we need to take the step further to bring the ability to decontaminate and clean and replenish these emergency vehicles closer to the problem, which is the trust. So we've developed this thing called what we call ARC, which is an ambulance readiness center, where the ambulance post the COVID positive patient or known infection patient could literally just go in you know, one way, get cleaned, decontaminated and replenished, and then go out, which would drastically save time and definitely fit with the whole infection prevention and control strategy. And then the same thing with air ambulance. We had a project that we've been working on to try and find a way of being able to decontaminate air ambulances, because when we had the chat initially, we've been told that they can't carry any COVID positive patients because they don't have a way of decontaminating the inside of the aircraft. So all of these things need to be considered, but it's important to know that the existing technology with a robust process could be proactively used to decontaminate all of those spaces. And we're absolutely happy to work with you guys and to adopt the best practice. Can I have the next slide, please, Freddie? I'll just wait for the next slide. And just finally, the topic that had been much discussed recently, and that's the PPE decontamination and of the reused disposable items. So globally, of course, there's been a shortage of personal protective equipment during this pandemic, and hospitals had to manage their strategies to deal with this. And a reactive strategy that has been employed in the US and had been approved by the FDA, and that was the hydrogen peroxide, the process that uses hydrogen peroxide vapor. It was quickly recognized in the UK that it could be one of the approaches, and we did some tests, and we strongly believe that is the right approach dealing with this crisis, if there's no other way other than to reuse PPE without the decontamination. And we previously shared the webinar, and there's a lot more detail on the previous webinar that we've shared with you guys, and there's a brief strategy when we think that, you know, combination of peroxide, peroxpod, and using a robust procedure that you could employ, you can proactively use the process to make sure that we have some stock of PPE that if in case we need to use it, it's there. And then we've shared the data that we got from Southampton trial, So we got some really good results to do with the decontamination of single-use gowns. So we've shared the data and we shared some instructions for what could be done locally in your trust to be able to benefit of this process. So anyone that wants to watch the webinar, you can find that on our website, and please do that and share that with anybody that's interested. And I suppose this summarizes our webinar now, and I think Freddie, we're going to go and take some questions.
Yeah, that's great. Thank you very much. I hope that was insightful for everybody, and yeah, we'd love to answer some questions if anybody has any. We open that up. Start us off, actually. I did receive a question earlier on in the week about somebody who was going to join us for the webinar today, asking about whether this segregation strategy should be adopted in other areas outside of a hospital environment. I wonder what your thoughts are on that.
Yeah, thanks. I think that's a good question. I think we're seeing hospitals adopt this methodology, particularly in the face of this COVID crisis. But I think we'll see any lessons and learnings we take from this current crisis will go off into the future and become general practice. I think also we'll see, well, just typically in the dentistry industry, they're being forced to adopt segregation methodologies, and I think it's putting them under amazing pressure, and I think it's something we need to be bear in mind. I think particularly for aerosol producing procedures or aerosol generating procedures, I should say, it's really putting those dentist facilities under massive challenge. I think we're seeing what was previously very much a hospital approach or a methodology or a strategy that was being previously very much isolated to hospitals, now becoming something that is being commonly used in other areas. Just the very principle of social distancing, I think, is an extension of an infection prevention and control practice. I don't know if that helps answer the question.
Yeah, I should think so. I'll have to feed that one back. Open to any other questions if anybody has any. Otherwise, Rick, I don't know if there's anything more that you'd like to elaborate on, or Tautis.
But just to follow on the question you've just posed, that I think the care home sector, which we all know have been hit quite hard during this pandemic, they definitely would be looking up to what the trusted NHS are doing, just to try and learn what proactive measures they can take in implementing in their setting to make sure that they can manage the risk of cross-infection, given the sensitivity and the prevalence in their environment. So to say that any of the points that we've discussed during this short, brief webinar, it can definitely be adopted across all of those sectors, including GP surgeries, etc., just to make sure that we manage all of these 4 vectors of transmission that we've just spoke about.
Okay, well, if we've got no more questions, I'll just let everybody know that we're actually going to be doing a series of webinars looking at these sort of ongoing, very topical items at the moment, and we'll be sharing those with you. And do keep an eye out, because we'll be looking to do those fairly frequently. We'll also share this webinar with you. We'll send that to your email address, and if you want to get in contact, please do. All our information is available on our website, and we're happy to hear, even just for a conversation with you. So thank you, everyone.
Just lastly on that last slide, Freddie, I think one thing I just wanted to emphasize, that we keep talking about life, we see it often in the press about life after coronavirus. I think what we're proving in healthcare is that, in hospitals particularly, that this is not life after coronavirus, this is life with coronavirus. And we're seeing hospitals set themselves up really for an expectation, a level of expectation around coronavirus going into the future. We had a very interesting conversation with somebody last week, and they were predicting that we are currently at around 15% through the current outbreak, the coronavirus outbreak. So that means we've got 85% still to run. Now, that might be a global view, I know, but nevertheless, that does give us some indication as to what level of work there is still to do in this. And I think we shouldn't be unmindful of the fact that there is likely to be ongoing risks and challenges and pressures on our healthcare facilities. I think we should celebrate the fact that infection prevention and control has had so much focus in both the public domain and by our politicians in the last few weeks. And I think we should use this as a great opportunity for us to establish infection prevention and control at the forefront of every healthcare facility. We are, as Freddie said, that we are running this as a series of webinars, and we're hoping to take you out to hospitals via these webinars. We can't do much by way of socialising at the moment, but we're hoping to take you out to these hospitals and show you real life experiences of where hospitals have implemented good practice and practices that we would recommend and commend to you. We'll be sharing them via these webinars. So I hope that we can use this to champion best practice. And if you've got any innovations, if you've got anything that you've done that you believe is best practice in healthcare generally, we'd love to hear from you. Please make contact. We'd love to hear. We'd love to share these good stories. So thank you from myself, and I'm sure Tautus and Freddie.
Thanks, Freddie, for hosting, and thanks, Rick, for your perfect insight and commentary. Thank you.
Yeah, thank you guys, everyone, for watching.
Thank you.